Uned-Derecho, video clase.
Hi everybody again. Finally, in this third lesson, we will talk about genome editing. That is a very interesting disruptive technology that's been very created. Well, indeed, this all started in 2012 when an article published in Science showed for the first time that cluster regulatory interspaces or palindromic repeats CRISPR. And take this word because it will be really important in the future, could be used as genome editing tool in a human cell culture, especially in conjunction with the Cas9 protein. Well, to talk in terms that you could understand, some scientists have discovered a kind of tool that made it possible for us to change the genome of a human being, of all human beings, and all beings in general, to be precise. Some years later, in 2018, a Chinese scientist, Dr. Jiankui, announced that the first modified human being had been born. Indeed, there were two twins, so we have at least 2 human beings that have already been modified in this world. A Russian scientist, Denis Rebrikov, is now proposing to create some new ones. So it seems that indeed we are in a moment where genome editing is becoming real. But it is very exciting from so many points of view, but it involves a number of ethical issues. I will not address here the editing of somatic line. I will not address the ethical issues involved in altering the kind of cells that we will not bring to our descendants, okay? Because this is not really especially relevant. It's very much connected with some other typical issues in clinical trials and research. Instead, I will focus in the changement made on the gene, on the genetic line. The kind of line that we are connecting with our descendants. Okay, so there are some arguments against German gene editing. Some of them are futility, risk, vulnerating the sacrality of the human genome, vulnerating human identity, and the slippery slope of eugenics, mostly. Okay, I will try to address most of them. Futility means that we don't need germline gene editing because we can do more or less the same things through pre-implantation genetic diagnosis. Well, this is true to a point. This is very useful as such to avoid that some people suffering from extreme genetic conditions come into this world. Yes, pre-implantation genetic diagnosis serves for this, but pre-implantation genetic diagnosis will never be able to help us with some of the most exciting promises of CRISPR-Cas, for example, trying to have a better condition as such. So I would say that they are not the same, and CRISPR-Cas is much more useful. Then we have the risk. Of course, we have an impressive risk at this moment if we use CRISPR-Cas in embryos. And indeed, one of the things that must be highlighted is that what the Chinese scientists did was really completely illegal and unethical because the risk was too high. So we all agree on the sense that risk should not be as high as it is right now to begin with this. But once this changes and the technology works better and we have a better knowledge of this technology, I think that risk will not be a key factor in terms of the ethical discussion. A more important factor here is what is usually called the vulneration of the sacrality of the human genome. Here some people oppose to germline editing because they state that it involves a definitive modification of the human genome, which is true or might not be true. It depends on what you are changing. If, for example, you are changing a kind of pathological expression of a gene related to Huntington disease for a healthy one, well, you are changing the genome of the embryo that is affected by this disease, but you are not changing as such the human genome. And in any case, it's not so important, in my opinion, whether you change the human genome or not. It's not something that is fixed. It continually changes, so it's very difficult to think about the sacrality of something that is continuously changing. I think that indeed, if we take this seriously, we should just do the opposite. I mean, we should start changing the human genome if we want to fix it. There's another thing that is a little bit different. Something people that consider that if we start editing embryos, we could change their identity. And this might also be true, but I don't think that this is necessarily wrong in the sense that identity, or our identity, is valuable, while it is not a terrible identity. But for example, if you are suffering a pathology that is causing harm to you, it might be part of your identity, but you could live better if you just rid of this kind of disease. So I don't think that this argument related to identity is a key argument against germline gene editing. Then we have a very, very well-known argument that is that, well, maybe some uses of germline gene editing related to healthcare and so on might be acceptable, but we should not provide it with green light, because otherwise this will bring us into a slippery slope to eugenics. I mean, this argument is a little bit pessimistic, because it thinks that if we start opening the door to CRISPR-Cas, we will end up with a kind of eugenic world quite similar to the world described in films such as Gattaca. But I don't think that this is a very, very strong argument, in the sense that usually we are able to put some cutting points to establish some boundaries, not to trespass, and sometimes works quite well. And I don't think that we should, just simply stop with all these just for this kind of fear that is not so rational, in my opinion. But then we have another arguments that are much more interesting, in my opinion. Those have to do with the social consequences. I think that they are powerful. One is that gene editing involves in vitro fecundation if we are to alter our descendants. So this means that only a few people will gain access to these technologies unless there is some public funding, which is hard to do. This means that people having money will be able to have better, healthier children. So there will be a kind of problems related to equity in using germline gene editing. And finally, we should think that if we go on with this, women will be somehow coerced to use IVF to be good mothers, because otherwise they will be just, letting the children be part of the gene lottery. That is precisely what CRISPR-Cas avoids. And that's all. I hope you have enjoyed this lesson, and thank you so much for your attention.